The nervous system is the master control system. It directs the activities of the body, maintains homeostasis and integrates incoming with outgoing information. It is the nervous system that is behind simple reflexes to complex patterns of thought, action, learning, and emotion. In this lecture, we will examine the general function and organization of the nervous system and the cells that make it work. The nervous system includes all neural tissue in the body. It receives, controls, integrates incoming information into appropriate responses. In order for it to carry out its role in the body, the nervous system has three overlapping functions. These are sensory, motor and integrative functions. The sensory component uses millions of sensory receptors of different types to monitor changes both inside and outside the body. These changes are called stimuli. The information gathered by sensory receptors is called sensory input and it is conveyed to the integration center. Here the appropriate motor output is sent to effectors such as glands, skeletal muscle, cardiac muscle or smooth muscle. In the example above, the thirst receptors convey information regarding dehydration status to the brain. The brain sends output along nerves to the biceps brachii and wrist flexors, and the person raises a glass of water to their mouth and drinks. The nervous system is an immense and intricate network, subdivided both functionally and anatomically. It consists of two primary anatomical divisions, the central nervous system CNS and the peripheral nervous system PNS. The central nervous system is composed of the brain, spinal cord, and, technically, the retina. It functions as the body's integrative and control center, responsible for processing sensory information, generating thoughts, coordinating movements, and regulating emotions and reflexes. In contrast, the peripheral nervous system includes all neural tissue outside the brain and spinal cord. This vast network connects the CNS to the rest of the body through 12 pairs of cranial nerves, which arise from the brain, and 31 pairs of spinal nerves, which emerge from the spinal cord. The PNS itself is further divided into sensory afferent and motor efferent divisions. The sensory, or afferent, division transmits information from sensory receptors to the CNS. These receptors detect stimuli from both the external environment, such as touch, temperature, sound, smell, and taste, and from within the body, including changes in blood pH, carbon dioxide levels, and the stretching of muscles or internal organs. This flow of information enables the CNS to interpret and respond appropriately to both conscious and subconscious stimuli. The motor, or efferent, division carries commands from the CNS to peripheral effectors such as muscles and glands. These effectors execute the body's responses, and the motor output occurs through two main systems, the somatic and the autonomic nervous systems. The somatic nervous system SNS controls voluntary actions such as skeletal muscle contractions, allowing for conscious movement and reflexive responses. The autonomic nervous system ANS, on the other hand, regulates involuntary processes. It transmits impulses from the CNS to cardiac muscle, smooth muscle, and glands, controlling activities such as heart rate, digestion, and glandular secretion. This system operates without conscious control, ensuring that vital bodily functions continue automatically. The ANS is further divided into two complementary branches, the sympathetic and parasympathetic divisions. The sympathetic division prepares the body for action, accelerating heart rate, increasing blood flow to muscles, and mobilizing energy stores, responses commonly described as fight or flight. The parasympathetic division, in contrast, conserves energy and promotes routine maintenance activities. It dominates during restful states, supporting digestion, recovery, and internal balance. Together, these divisions of the nervous system maintain communication throughout the body, integrate sensory input with motor output, and preserve homeostasis across all physiological systems. Even though the nervous system is extremely complex, there are only two main types of cells. Neurons are the structural functional unit of the nervous system. They receive stimuli, process it and if appropriate generate an action potential. The neuroglia, which literally means neuron glue because they had no clue what it did at the time, are supportive cells. They protect and support the neurons physically and physiologically. They also outnumber neurons by 10 to 1. Another interesting thing about neuroglia is that they never lose their ability to divide as do neurons. Consequently, most brain tumors are gliomas or tumors formed from neuroglia. Neuroglia are found in both the CNS and PNS. We will discuss the function of the main types of neuroglia found in both subdivisions of the nervous system. The neuroglia of the CNS includes astrocytes, oligodendrocytes, microglia and ependymal cells. Of all the neuroglia, astrocytes are the most numerous. They are indispensable to the normal function of the neurons. Astrocytes get their name star cell from their shape. They have many processes that emanate out from the center of the cell. Astrocytes assist in metabolism of the neurotransmitters that neurons make to send signals to other neurons or various effectors. They help to maintain proper potassium levels so that neurons can generate action potentials. During development, astrocytes guide neurons to their proper contacts and location. Assist in creating the blood-brain barrier. Physically intertwine themselves around neurons to support them. Provide a means of obtaining nutrients and getting rid of waste by connecting with blood vessels. This diagram shows how the astrocyte prescreens materials coming from the bloodstream. We will learn in the lecture on the brain, that there is a barrier between the blood and the interstitial environment of the neurons. To ensure proper function of the neurons, the astrocytes tap into the blood capillaries and select only the appropriate ions and molecules from the bloodstream, and convey these to the neuron. The astrocyte also picks up the metabolic waste produced by neurons and transports that material to the capillaries where it is carried away in the blood. The microglia are small phagocytic cells derived from circulating blood monocytes. Recall that monocytes are a type of white blood cell that function in immune responses. They also are the source of all tissue macrophages. The microglia help destroy invading pathogenic organisms or clear out cellular debris resulting from cell death. The name oligodendrocyte means few processes cell or a cell that has several branches. The number of oligodendrocytes increases with increasing complexity of the nervous system. The human nervous system has the highest number of oligodendrocytes/neuron compared to those of other vertebrates. Oligodendrocytes produce the insulating myelin sheaths that surround axons in the CNS. We will discuss the process of myelination later on in the presentation, but this picture shows an oligodendrocyte and its processes wrapping around small segments of several nerve fibers. The nerve fibers are the axons of neurons and are seen above in yellow. An oligodendrocyte can be involved in myelinating small segments of 3 to 50 different axons. Ependymal cells are ciliated cells that line spaces in the brain called ventricles, and the central canal of the spinal cord. The beating of their cilia helps to circulate cerebrospinal fluid that fills the cavities, forming a protective cushion both inside and outside the central nervous system. The small picture at the bottom of the slide is a histological section through the central canal of the spinal cord. CSF is shown in the canal, which is lined with the dark staining ependymal cells. This diagram illustrates the relationship of the CNS neuroglia with neurons. The supporting cells of the PNS are of two types. The neurolimocyte, also known as a Schwann cell, and satellite cells. The neurolimocyte or Schwann cell produces the myelin sheath surrounding axons of the PNS. Wherever a Schwann cell covers an axon, the outer surface of the Schwann cell is called a neurolima. It contains the nucleus and the outermost cell membrane containing myelin. Instead of having several processes that wrap around small segments of several axons, the neurolimocytes wrap themselves around and around a segment of an axon, creating the myelin sheath. Satellite cells surround and support ganglia, which are clusters of neuron cell bodies occurring in the PNS. The satellite cells regulate the environment around the neuron cell bodies within the ganglion. In the picture above, the purple cells surrounding the large yellow cell body of a neuron are satellite cells. Below is a micrograph of a histological section through a ganglion showing about a dozen large round D cell bodies surrounded by small dark staining satellite cells indicated by the red arrows. In the PNS, each Schwann cell wraps around about a 1 mm length of an axon and spirals around it many times. Most axons are surrounded by an insulating layer of lipid combined with protein called myelin. The myelin sheath functions to electrically insulate the axon. This greatly increases the speed of conduction of nerve impulses. The amount of myelination increases from birth through adulthood. In unmyelinated axons, the impulse must travel continuously the entire length of the axon. The plasma membrane of the axon must depolarize in a continuous wave. Depolarization is a flip-flop of positive and negative charge on the inside and outside of the axon membrane. Without a myelin sheath, Conduction of the action potential is continuous down the entire length of the axon. It is analogous to walking heel to toe. Along a myelinated axon, there are nodes that lack myelin. Essentially, the action potential jumps from node to node, skipping over the myelinated segments of the axon. This greatly speeds up conduction. This type of conduction is called saltatory conduction. To review, myelination in the CNS and PNS is achieved by different types of neuroglia. In the CNS, oligodendrocytes send out branches that can myelinate segments of 3 to 50 axons. In the PNS, Schwann cells or neurolimocytes myelinate a segment of a single axon. Multiple sclerosis is the most common of a group of devastating demyelinating diseases. MS is an autoimmune disorder in which cells of the immune system fail to recognize the myelin itself, and begin attacking it. This results in inflammation which causes scarring sclerosis around the nerves. The scarring slows or blocks nerve impulses, resulting in decreased muscle coordination and strength, and disturbances of sensation and vision. Neurons, also known as nerve cells, are highly specialized both structurally and functionally. Their primary role is to generate and transmit information in the form of nerve impulses, or action potentials, from one region of the body to another. In the image above, the single blue neuron is shown forming a connection with part of a purple neuron. The neuron that sends the electrical message is called the presynaptic cell, while the neuron or other effector that receives the signal is known as the postsynaptic cell. Although neurons vary widely in their shape and structure, all share three essential components: the cell body, dendrites, and axon. The cell body, also called the soma or pericryon, houses the nucleus and most of the neuron's organelles, including the cytoplasm, Golgi apparatus, rough endoplasmic reticulum, mitochondria, and cytoskeletal structures such as neurofilaments and neurotubules. The RA is especially well developed in neurons because of their high demand for protein synthesis, particularly for neurotransmitter production. Under the light microscope, this RA appears as dense, granular regions known as Nissl bodies, or chromophilic substance. Extending outward from the cell body are the dendrites, which serve as the neuron's receptive surfaces. Dendrites often form extensive branching structures, known as arborizations, that greatly expand the area available for synaptic connections. This branching allows a single neuron to receive and integrate information from the axons of many other neurons. Along their length, dendrites possess numerous small projections called dendritic spines, which are the primary sites of synaptic input. In fact, approximately 80 to 90% of a neuron's total surface area consists of these dendritic spines. Projecting from the cell body is a single, often long, axon, also referred to as a nerve fiber. The axon is responsible for conducting electrical impulses away from the cell body and transmitting them to the postsynaptic cell. Because of the axon's specialized structure and electrical function, specific terminology is used to describe its regions. The axoplasm refers to the cytoplasm within the axon, while the axolemma designates its plasma membrane. The axon hillock is a cone-shaped region that marks the transition between the cell body and the beginning of the axon. Just beyond this lies the initial segment, a crucial area of the neuron where the action potential is first generated. Together, these structural components enable neurons to function as the communication network of the nervous system, efficiently transmitting electrical and chemical messages that underlie thought, sensation, and movement. Each neuron possesses a single axon, which serves as the pathway for transmitting nerve impulses away from the cell body. Although every neuron has only one axon, the length of this process varies greatly depending on the type and location of the neuron. For example, axons belonging to motor neurons that originate in the spinal cord and extend to muscles in the foot may reach lengths of up to 1 meter, or approximately 40 inches. The axon emerges from a specialized region of the cell body known as the axon hillock, a small raised area that lacks Nissl bodies and therefore appears pale or clear under a light microscope. Immediately following this region is the initial segment, which represents the first portion of the axon. It is here that nerve impulses, or action potentials, are generated before propagating along the remainder of the axon's length. Within the axon are numerous mitochondria and cytoskeletal components, including neurofibrils and microtubules, which provide structural support and enable intracellular transport. These microtubules play an essential role in moving vesicles filled with neurotransmitters toward the axon terminals, where communication with other cells occurs. As the axon approaches its target, it may branch into smaller extensions known as collaterals or teledendria, allowing the neuron to form multiple synaptic connections. The terminal ends of these branches, called synaptic terminals, are the specialized sites where neurotransmitters are released to convey signals to the postsynaptic cell, whether another neuron, a muscle fiber, or a gland cell. A synapse is defined as the site of communication between two excitable cells, a neuron and another cell neuron, muscle, gland, other cell types. Most synapses are chemical or electrical. The presynaptic cell of a chemical synapse makes vesicles of neurotransmitter and transport them from the cell body to the axon terminal. Movement of synaptic vesicles in this manner is called axoplasmic transport. The packets of neurotransmitter are released into the synaptic cleft once the wave of depolarization, i.e., the action potential, reaches the end of the axon synaptic knob. The neurotransmitters fuse with their receptors on the postsynaptic membrane, thereby conveying the message. On a clinical note, axoplasmic transport occurs in both directions. Transport from the cell body to the synaptic knob is anterograde forward plus flow. Transport in the opposite direction from synaptic knob to cell body is termed retrograde. Rabies virus, injected into peripheral tissues via a bite from a rabid animal, enters the synaptic knobs quickly and viral particles are transported to the cell body, which lies in the CNS. Kills CNS neurons and is fatal. Similarly, many toxins, heavy metals, some pathogenic bacteria and other viruses bypass the CNS protective defense mechanisms by undamaged cell bodies via retrograde axoplasmic transport. As mentioned previously, neurons come in many sizes and shapes, and are therefore divided into structural classes on the basis of the relationship of the dendrites to the cell body and axon. Anaxonic neurons are small neurons, with no visible distinction between axons and dendrites. All cell processes look alike. Located in brain and special sense organs. Function is poorly understood. Bipolar neurons have two distinct processes. One dendrite that branches extensively and one axon. The cell body is located in between. Bipolar neurons occur in special sense organs. The special senses, sight, smell, and hearing, are relayed from sensory receptors to other neurons via bipolar neurons. Bipolar neurons are small, their maximum size is 30 mm. Unipolar neurons are also referred to as pseudounipolar because the dendrite and axon are continuous fused and the cell body lies off to one side. Unipolar neurons are the sensory neurons of the PNS. Axons may extend over 1 meter toes to brain. Multipolar neurons have two or more dendrites and a single axon. This structural class includes the motor neurons that control skeletal muscles. The axons can be extremely long, brain to toes. Multipolar neurons are the most common neuron type, comprising more than 99% of neurons in the human CNS. Sensory or afferent neurons conduct impulses from sensory receptors in the skin, muscle, or viscera to the CNS. Their cell bodies lie in ganglia in the PNS. There are approximately 10 million sensory neurons in our nervous system. What distinguishes what kind of sensation sensory neurons can convey depends on the receptor. There are three main categories of sensory receptors. Interoceptors monitor the digestive, respiratory, cardiovascular, urinary, and reproductive systems, provide sensations of taste, deep pressure, and pain. Exteroreceptors provide information about the external environment: touch, temperature, pressure, sight, smell, and hearing. Proprioceptors monitor the position and movement of skeletal muscles and joints. Motor or efferent neurons transmit impulses from the CNS, brain, or spinal cord to effectors in the body periphery. There are approximately 500,000 motor neurons. Somatic motor neurons innervate skeletal muscle, and visceral motor neurons, autonomic neurons, innervate all peripheral effectors other than skeletal muscle. Association neurons, interneurons, complete the communication pathway between sensory and motor neurons. Their cell bodies lie in the CNS. There are approximately outnumber all other types of neurons combined. They distribute sensory information to the appropriate region of the brain or spinal cord, and coordinate motor activity. One or more interneurons are situated between sensory neurons and motor neurons. The more complex the function, the more interneurons are involved. Compare a reflex to a higher order function such as memory, planning, or learning. Neurons have a very limited ability to regenerate if injured. Structurally too complex to replicate or repair. Very complex interactions with other neurons. Beginning before birth until about 6 months of age, all developing neurons have lost their ability to undergo mitosis. Once destroyed, they can't be replaced. In the CNS, repair is practically non-existent. In the PNS, some repair is possible under certain conditions. The damaged neurons are part of a myelinated nerve, and the neurolymphcytes Schwann cells are active and intact. If the cell body soma is undamaged. When an axon of the PNS is injured, the nucleus migrates to the periphery and Nissl bodies break down in a process called chromatolysis. The axon distal to the site of injury degenerates along with its myelin sheath. This is called Wallerian degeneration. Macrophages are drawn to the site of degeneration and remove cellular debris. A portion of the axon proximal to the injury site degenerates, but only to the first node of Ranvier. Regeneration of an injured axon involves synthesis of new Nissl bodies RR, as well as mRNA and RNA so that proteins can be synthesized. Although the axon and its myelin sheath degenerated, the neurolemma remain intact. This is important because without the neurolemma the new regenerating axon could not grow back exactly to the appropriate receptor or effector with which it formed the synapse. Neurolemmacytes on either side of the injury begin to multiply mitotically and form a tube that bridges across the injured area. This tube guides the regenerating axon exactly back to its site of contact with the receptor or effector. Axons grow at a rate of about 1 to 1.5 mm/day.